Welcome. I am Meg Smith, CEO of Cure Alzheimer's Fund. Thank you for joining us today to hear about how we are advancing the fight against Alzheimer's disease. Cure Alzheimer's Fund has one mission: to fund research with the highest probability of preventing, slowing, or reversing Alzheimer's disease. Since 2004, we have deployed more than $210 million toward extraordinary research to end the terrible burden of this disease. Active researchers from a wide range of scientific specialties who are at the forefront of discovery guide our funding while also pursuing their own breakthrough projects with our support. At Cure-Alls, we focus only on making bold bets to generate knowledge that will help the entire Alzheimer's community. We do not pursue profits or financial returns. We can provide this support to the most innovative ideas across biomedical science, prioritizing new, neglected, and unfunded questions fundamental to understanding what causes and contributes to Alzheimer's, only because of the generosity of our donors. Our board of directors, trustees, and certain other supporters cover all of our operating costs. This generosity and vision mean that 100% of the gifts from our other wonderful donors go directly to our research program. We are grateful for the community of generous donors who make these scientific advances possible, and I am glad to share some of them with you today. After watching, I hope you feel as I do: incredible resolve to find a cure for everyone touched by this terrible disease.
I think there's no exaggeration in saying that there's no foundation like Cure Alzheimer's Fund when it comes to funding the highest impact, most meaningful research that has changed the field for 20 years and continues to do so. No foundation comes close to comparing to Cure Alzheimer's Fund. And that's because we really choose the best people in the world, the best projects. We have a very, very rigorous vetting process for what we fund, and our focus is always on funding grants and projects that are going to push the ball forward to someday have a cure to end this disease. And today, I'm going to talk to you about our progress in doing just that. I'll be talking today about the hope that's on the horizon, and at the end, what can we do now to lower our risk. I want to start as obligatory from Harvard and Mass General. These are my disclosures over the past years regarding consulting fees, financial interests, honoraria, and royalties. So first, I just want to show something that to me, it just always blows my mind. If you look at the age distribution in 1960, you see a classic pyramid, right? You see many younger people, and by the time you get to older age, over 60, you start to narrow down and you have a pyramid. But by 2060, it's going to be more of a pillar. It's going to look like this. And that's because the good news is that thanks to modern medicine and healthcare, the average lifespan in the USA is already around 80 years. And the fastest growing sector in the United States in terms of age is over 85 years old. The bad news is that despite this lengthening lifespan that we can all enjoy, there's a growing gap between our lifespan and our health span. And this is especially true for our brain health span. We are far outliving the longevity of our brain health. This is not as true for the organs under the neck, where we take care of them on a daily basis and yearly basis. But it's especially true for our brains. And on top of that, over the age of 85, this growing sector, fastest growing sector, 33% of those over 85 already show the clinical symptoms of cognitive impairment, most of that due to Alzheimer's disease type dementia. And this is also bad news that, over 20 years from 2000 to 2022, 22 years, you can see that diseases like breast cancer, prostate cancer, heart disease, stroke, HIV, all have been stabilized or are down. Alzheimer's, on the other hand, went up 142%. Why? We're growing older. And as we grow older, this disease increases with age. So here's some facts about Alzheimer's I just want to begin with. It is the most common form of dementia in the elderly. People ask me the difference often between dementia and Alzheimer's. Dementia is an umbrella term, like fruits. And one of the most common fruits is apples, okay? So Alzheimer's is the most common dementia. And it's about 80% of cases of dementia, followed by cardiovascular or neurovascular dementia and frontal lobe dementia. We have 7.2 million patients in the US, 32 million worldwide. This will double every 20 years. And in terms of prevalence, 1 in 9 people over 65 have it. 5% from 65 to 74, 13%, and so on, up to 33% over 85. Now, one of the things I'm really going to emphasize here is that Alzheimer's brain pathology, in other words, biological Alzheimer's disease, begins on average 22 years before there's any symptoms. So this gives us a window for early detection, early intervention. And this is truly how I think of how we're going to beat this disease. And I'm going to spend a lot of time on that in my presentation. Risk factors, of course, age is number one, family history, stroke, brain injury, sex, meaning 2/3 of patients are female. It's really a disease that strikes women more than men. Some of the midlife risk factors include obesity, having high cholesterol, hypertension, diabetes. And given that these diseases are also so prevalent in our society today, if we look at the Alzheimer's disease cost in the USA right now, it's $384 billion per year. This takes up 64% of the Medicare Medicaid budget. And given that the number of cases will more than triple by 2050 to at least 25 million, or more, this will basically collapse Medicare and Medicaid. So we really have to do something about stopping this disease. Now, people often ask me the difference between a senior moment and getting Alzheimer's. And these are just some senior moment jokes that I found. We successfully moved a word that was on the tip of your tongue. Oh, crap, was that today? And then this guy looking at the side, feasible reasons you may have had for walking into this room. Look, we all do that. We walk into a room, forget why. We forget who we're calling on the phone. Thank God cell phones show us that now. But here's how to think about the difference between age-related changes and Alzheimer's changes. I can't go through all of these, but for example, forgetting names is common as you get older, especially over 50 or 60. But when memory loss is disruptive to your everyday life, it's different. Forgetting to pay a bill, fine, but you can't follow instructions anymore. You forgot how to set your DVR or the cable box. Well, forgetting how to actually drive to work or drive back home, that's what you worry about. Forgetting the day of the week, fine, but if you totally lose track of the month and a year. So on and on and on, and you can just take a look at these. The bottom line is that when something is no longer a senior moment when it's repetitive and and it's a routine thing that disrupts your everyday quality of life. And then then you have to start thinking whether it might be Alzheimer's. And there are also reversible or temporary causes of the symptoms of dementia. For example, depression, being extremely stressed, not getting enough sleep or sleep apnea. You could have a delirium due to a urinary tract infection that requires medical treatment. Side effects of medications, Lyme disease, thyroid problems, a head bang with a concussion. It can also be due to a blood clot or tumor in the brain that might be need medical attention. That one not so reversible. Vitamin deficiencies like vitamin B12, folate, omega-3, and excessive alcohol consumption can also lead to these types of symptoms that in this case, you can do something about. But if it's Alzheimer's disease, you see this pathology that was first described in 1906, okay? Beta amyloid deposition or plaques, shown here. Tau tangles, which are inside the neurons here, blocking the function of the neuron, and neuroinflammation. I'll get more into these as we go along. But this is probably the most important slide you'll see, or among the most important you'll see today, about how this disease stages itself. First, amyloid deposits in the brain, the sticky material made up of this protein called the amyloid beta protein. For short, we call it A-beta. When the amyloid beta or A-beta starts to stick together, you get these sticky plaques outside of neurons. And these deposits in the A-beta clumps, or as we call them, oligomers, can drive the formation of these tangles inside of nerve cells, tau tangles or neurofibrillary tangles. And this causes a certain amount of cell death, but actually not quite enough to give you the disease yet. I like to say the amyloid's like the match, and the tangles are like the brush fires, and they spread and propagate in the brain once you make them. So they're like brush fires that spread. But once this process leads to neuroinflammation, now you have the wildfire. Now you start to see symptoms. Neuroinflammation is a response of cells in the brain called glial cells, which are normally like housekeepers. But if they sense something's wrong, like neurons are dying, or there's all this sticky material around, like amyloid, they can convert from the from nurturing and housekeeping in the brain to thinking they have to defend the brain against an infection. If neurons are dying in the brain, these little microglial cells, they assume it's an infection. It's perceived that way. And now they start to destroy that part of the brain so this perceived infection doesn't spread. That's what neuroinflammation is. And that's when it really, the brain goes downhill and you start to get symptoms of dementia at this point. And this whole process can take up to 20 to 30 years. But because it's a slow process, it gives us a window of opportunity for early detection, early prevention. And that's what we do, right? With diabetes, heart disease, and many cancers, we detect things early below the neck and we stop them before they become a problem, right? We don't start giving you a statin for cholesterol only when you have congestive heart failure, right? But for Alzheimer's and other neurodegenerative disorders, we don't do that yet. But the fact is, for Alzheimer's, we can now. We can detect this disease early. We don't have to wait to clinically diagnose this disease only when the brain has already undergone major damage. So I'll get more into that. So the way we're going to beat this disease, and this was the original mantra of Cure Alzheimer's Fund 20 years ago when we first started this foundation: early prediction of the disease, followed by early detection of the pathology, which as I said, begins decades before the symptoms, and then early intervention, stop that pathology before the onset of the symptoms. So let's go through early prediction first. The first three genes that I and others found back when I was a student at Harvard, and then a little bit later when I first started as faculty at Mass General, were the early onset genes: the amyloid precursor protein APP, presenilin 1, presenilin 2. There are over 200 mutations in these genes that guarantee the disease. Okay, it doesn't matter what I'm going to tell you about lifestyle. These mutations guarantee the disease under 60 years old, often under 50. It's only 1 to 2% of Alzheimer's, but the most severe form. And every one of these mutations do the same thing. They all increase the formation of amyloid in the brain, so you make amyloid in your brain faster over your lifetime than when you have the mutations in these genes. The other major gene is ApoE or ApoE. This is a late onset gene, and a variant of this gene, ApoE epsilon 4, is present in about 25% of the population, but it's in 50 to 60% of Alzheimer patients. And when you inherit one copy of the C4 variant from a parent, it increases your risk by 3 or 4-fold. If you inherit 2 copies of E4, which is less likely, but about 2% of cases, then you have a 14-fold increased risk. And again, what does ApoE4 do? It makes more amyloid form in the brain faster because it decreases the amyloid clearance from the brain. These mutations increase the production of the amyloid in the brain. Production up, clearance down. But amyloid is the key to all 4 of these first Alzheimer's genes we knew. Now, since then, we've done a lot of work, including the Alzheimer's Genome Project. This is a paper that's in press right now where there are many co-authors on this paper, including myself, that shows the update on how many Alzheimer's genes we now know. There are over 100 validated Alzheimer's genes because we can do what's called genome-wide association studies, scanning the whole genome at once. And as you may or may not know, it was Cure Alzheimer's Fund who gave the funding to my lab for the very first genome-wide association screen when this technology first came around in 2005, 2006. And that's how we found one of the most important genes today in Alzheimer's, which is called CD33, which drove the field to microglia. And I'm going to talk a lot about microglia as a target for stopping this disease later. CD33 was the first, and that was thanks to Cure Alzheimer's Fund. And now we have many, many more genes. I don't have time, of course, to go into all of them here. And I just want to also say that we're finding more genes all the time. That's another study funded by Cure Alzheimer's Fund, where we looked at populations of mixed ancestry, so ethnically diverse populations. And by doing this, we found 16 new genes that you would not have found if you looked at your typical population sample of Caucasian European descent individuals. Here we looked at Hispanics, we looked at African Americans, And by looking at multi-ancestry, we found 16 new genes that otherwise would not be found, which emphasizes the need to include diversity in research studies. So how do these genes work? Well, here on the y-axis, going up, there's increased probability of getting the disease if you inherit the gene mutation or the genetic strength. On the x-axis, going this way, is how frequent is that mutation? How common is it, right? So the early onset genes are very rare, but they guarantee the disease. Then we have, like I just showed you, 100 or so genes that are have very common variants in them, but have a low effect on whether you're going to get the disease. And then you have ApoE, the big daddy here, where, as I said, one copy of E4 increases risk fourfold, two copies 14-fold. This is in 60% of patients have one E4, and about 11% of Alzheimer's patients have two E4s. And many of you listening today may have had your ApoE genotype done. I can just tell you that inheriting ApoE does not guarantee the disease. It increases risk. And the good news is this, except for these 1 or 2% of mutations, 1 or 2% of cases with mutations in these three genes, for all these other genes, which is 98 to 99% of Alzheimer's genetic risk, lifestyle matters. Lifestyle can offset risk for all these other genes, including ApoE4. So at the end, we're going to talk about what you can do about lifestyle. Another thing that Cure Alzheimer's funded was a polyomic risk score. So you might have heard of a polygenic risk score. In other words, if you take all the Alzheimer's genes together, what is your risk? More recently, we were able to add to genomics metabolites or metabolomics, proteins or proteomics, take into account which prescription drugs you took over your lifetime. And using all this information with very sophisticated algorithms, we came up with a polyomic risk score to predict your risk. So this is all about how to predict your risk for disease. Of course, the easiest way to predict your risk is look at your family. Family history is a big predictor of disease risk. So once we have early prediction, and let's say you do have a family history, if you have a mom, dad, first-degree relative who got this disease, take the earliest age of onset in your family, and then subtract 30 years from that. And that's when, if you are worried about yourself, you might think about early detection. Why? Because we can detect this pathology in the brain 20 to 30 years before symptoms. And when you should have a test for early detection depends on your family history and age of onset, to subtract the 30 years from the youngest onset in your family and a first-degree relative. So what do we do for early detection? Well, here's the staging of the disease, okay? So very early, the amyloid accumulates, and we can see that with a blood test. There's now a blood test that can tell us whether you have amyloid in your brain. You can look at what's called the A beta 42 to 40 ratio, or you can look at this marker called p-tau 217. Even though this is a marker for tangles, p-tau 217 tells you if you have tangles in your brain, specifically because you have amyloid in your brain. Amyloid-induced tangles. You can have boxed your whole life or played a lot of football and have tangles for other reasons. That would be a different tangle marker like p-tau 181. So these are the tests that can tell you if you already have amyloid accumulating in your brain. They are already available. And then later on, there are biomarkers that tell you how much neurodegeneration, how many neurons are dying, which is the result mainly of neuroinflammation, that wildfire I showed you early. And here you have NFL, or neurofilament light chain, as a biomarker. Now let's do the staging of this disease, because now we have stage 0, means nothing's happening yet. You have genetic risk. You might carry 2 ApoEs and have that 14-fold increased risk, or an early onset gene mutation that guarantees the disease. Stage A, you have amyloid accumulating in the brain. Not much tangle yet, but amyloid. And then stage B, now you have amyloid, the tangles are forming, neurons are starting to die. This is triggering neurodegeneration. Now the earliest symptoms are starting to show up. That's stage B and C. And by stage D, where you have mild to moderate to severe, you have a lot of amyloid tangles, and neuroinflammation is causing nerve cell death and loss of synapses, the loss of connections between nerve cells. Remember, you have 100 billion nerve cells in your brain, trillions of synapses or connections making up your neural network. At the end of the day, it's loss of synapses that correlates most with degree of dementia. So this is the course of this disease. So recently there was a survey done. Would you want to know if you have Alzheimer's pathology in your brain pre-symptomatically? Are you one of these people who are, already forming plaques and tangles in your brain, but you're still a decade or two or more away from symptoms? So we asked that. 91% said yes, they'd want to know. I'm a wimp. I wouldn't want to know. I'd want to live in ignorant bliss. But a lot of people just would rather know what they're up against. So 91% said yes. And here we have, they could find out. You could go to a company like C2N or another one that looks at your plasma amyloid beta and p-tau217 and tells you whether you have amyloid in your brain. Now, the thing is, these blood tests are not covered by insurance. They wouldn't be covered. It's about $1,500 to have one of these tests because the insurance companies say they're not actionable, meaning if you have a positive test, there's not something they can tell you can do right now that's covered by your insurance or Medicare or Medicaid that will allow you to prevent the disease. In other words, early prevention. So you have to pay out of pocket if you want to find out. So what about early intervention? Well, the key, as I told you, the key pathologies that begin this disease, initiate it or trigger it, are amyloid or A beta and tangles or tau, right? So if you want to target A beta, there's all different ways. You can turn off the APP. That's the gene that makes the A beta. You can try to block the enzymes that cleave to create the A beta when it cleaves to amyloid precursor protein. You can prevent the aggregation of A beta. You can try to clear the plaque out. And likewise for tangles, you can try to block the tau, stop the modification of tau that leads to tangles. You can try monoclonal antibodies that prevent the spread of the tangles. So these are the different ways to do it. I'm going to focus on amyloid because it's very clear now from longitudinal brain imaging studies, the faster you accumulate brain amyloid, the sooner you get AD. It's very, very similar to cholesterol and heart disease, right? The faster you accumulate cholesterol and plaque in your arteries, the sooner you get coronary artery disease or congestive heart failure. And very important, if you have congestive heart failure, would your doctor say, oh, we'll just lower your cholesterol and you'll be fine. Take a statin like Lipitor. No, because you had to do that 20, 30, 40 years ago to avoid getting to the point of congestive heart failure. Amyloid's exactly the same. Treating amyloid and getting rid of it is something you need to do as early as possible, preferably someday for prevention, secondary or primary prevention. If you already have the disease, you have symptoms of dementia, there's tangles, there's neuroinflammation. Remember, the amyloid was the match. You're blowing out a match when there's already a wildfire. And that's why there was so much controversy over the years over amyloid, because by simply removing amyloid wasn't enough. Now, more recently, as I'll tell you, there were drugs that remove the amyloid that lead to some cognitive stabilization if you do it very early. And those drugs were approved. We'll get to those in a minute. But I like to use a kitchen sink as an analogy for amyloid in the brain. Here's a kitchen sink overflowing with water, like the brain overflowing with amyloid deposits, okay? So if you have a kitchen sink overflowing with water, what do? Well, two things. You try to turn off the tap, or you try to unclog the drain and get rid of it. The two newest approved Alzheimer drugs, Leqembi and Kisunla, are monoclonal antibodies that bind amyloid and trigger its removal in the brain. So that's on this side. Meanwhile, for stopping production, I'll tell you about a drug that my lab has been developing for 25 years now, that Cure Alzheimer's Fund has been instrumental in funding and bringing to clinical trials hopefully soon. So here's these new approved monoclonal antibodies, Leqembi from Eisai, Kisunla from Lilly. They've only been approved for the earliest stage Alzheimer patients because that's what the trial was done in. And in those earliest stage patients, these antibodies very nicely clear amyloid from the brain over 6 months to a year, maybe 2 years. And they were shown to slow down the cognitive decline. So they didn't make people cognitively better, but they slowed down their decline of their cognition by about 25, 30%. And that was taken to validate the amyloid hypothesis, but it still said, you know what, it's still too late. Treating amyloid after symptoms of dementia, like I said, it's like using cholesterol drugs after congestive heart failure. If you get there really early, there's some help, but you really want to be able to do this. We need to be able to treat people who have the blood test and they find out they're amyloid positive, and this is when you want to try to treat them. Just like if you find out you have high cholesterol, you take a statin early in life. So if everybody had the blood test to say, do I have amyloid in my brain, but I don't have Alzheimer's symptoms yet? Am I on my way? On the low end, 10 million. On the high end, probably 40 or 50 million Americans would find out they already have biological aspects of Alzheimer's, meaning the amyloid and tangles are already starting to accrue in the brain. And what we want to do is give them affordable and safe therapies to avoid the progression from just pathology positive to cognitive impairment and dementia. That's our goal. So there are prevention trials going now with the amyloid antibodies. These are all the different prevention trials, many of them happening at Mass General Brigham at Harvard, and we're waiting to see how they do. But the problem is this: these antibodies, they're really expensive because they have side effects that require monthly MRIs to make sure you don't have any brain swelling or edema, to make sure you don't have any microhemorrhages. Now, these side effects, as scary as they sound, are reversible. They're very common, but they're reversible. So if the MRI shows there's an issue, you stop and you reverse it. But it makes for very expensive treatment that has these side effects. So as I said earlier, we need safer, more affordable alternatives to manage our brain amyloid later in life and stave off this disease. So in terms of a little white pill that might do that, my colleague Steve Wagner, he passed away a few years ago, but 25 years ago, he and I invented a class of drugs called gamma secretase modulators. And today, if you ask across the field, what is our greatest hope for a little white pill that might lower the production of amyloid? Universally, everyone will say gamma secretase modulators. It's taken 25 years to develop these drugs. Cure Alzheimer's Fund has been amazing in terms of support of development of these drugs, as has been the NIH and the Neurotherapeutics Blueprint program of the NIH that Francis Collins had started. And thanks to all of that hard work, now we've taken our gamma secretase modulator, our best one. The NIH said, look, start a company to do this. You don't want to be an academic putting a new class of drug in a person. So I started a company called Active Pharmaceuticals, in which I have an equity stake back in 2022. They licensed the intellectual property for these drugs from Mass General. And we're hoping to get our first phase 1 clinical trial going maybe this fall or early next year. We're waiting for the FDA now to tell us whether we can proceed. So we put in our what's called IND, Investigation of New Drug Application. We're waiting to hear. And hopefully, thanks to Cure Alzheimer's Fund and all the support we have from them, this new class of drug that's our greatest hope for as a little white pill, like Lipitor is used for cholesterol, right? This would be like the statin for Alzheimer's if it goes the way we want. A little white pill that prevents the amyloid production early on so you avoid the disease, just like trying to avoid heart disease by managing your cholesterol. This is a great hope for us. And I should also say that for that 1 or 2% of people who have early onset familial Alzheimer's due to those 3 genes that guarantee the disease, APP, presenilin-1, presenilin-2, this drug stops that. This drug will, if you know you have that gene and it's a death sentence, you know you're going to, that mutation guarantees you're going to have the disease by 60 years old, this drug will reverse it. So this is hope for people who otherwise had no hope. So never mind as a statin for everyone. For early onset disease, with that guarantee of the disease, you start taking this drug early enough in life and it reverses what those early onset mutations do to guarantee the disease. And that's also very exciting. So we're hoping the FDA lets us start our trial. So everybody keep your fingers crossed for that. So what else can we do to come up with new drugs? And recently what we've been doing is trying to learn from super ages centenarians. A super ages centenarian is someone who's over 100 years old with exceptional cognitive ability. Like here, Ms. Senhouse from Wellesley, Mass., who died at 112 in 2024. Right? This just shows you how many centenarians there are. It's about 1 in 5,000 people, 201 million. Semi-super centenarians, 105 to 109. Super centenarians, over 110. There's only about a very few of those. Cognitive super ages are who we're focusing on, and cognitive super ages centenarians, very rare. one or fewer in 1 million or over 100 who still have exceptional cognitive ability as if they were still 40 or 50. And the reason for this is there's two reasons how you can be a super ager centenarian. You can have resistance in the brain, meaning for some reason you hit 110 and you have no amyloid and no tangles, or it's very low. That's called resistance. Resilience is different. Resilience means you made amyloid, the match was lit, you have the tangles, the brush fires are spreading, but there's never a wildfire. It never turned into neuroinflammation, so you stayed cognitively intact. Resilience versus resistance. So what confers resilience and resistance? I mentioned this cell type in the brain earlier, microglia. So microglia are housekeepers that can clear amyloid for you and get rid of it. And if they do a really good job, they can confer resistance. You don't make amyloid for most of your life, or like a centenarian, even over 100. But they're also sentinels. As they're clearing, as they're housekeeping, they're keeping track. They're saying, am I eating microbes? Because that might mean there's an infection. Or am I eating dead cells? Which I also perceive there might be an infection, whether there's one or not. Either eating up a microbe while they're cleaning or a dead cell starts to trigger them from changing from housekeepers to killers. Now they're saying, oh, wait a minute, cells are dying. Or worse, I just ate a virus. Now I'm activated to wipe out this part of the brain because the brain's enclosed and I can't let this infection spread. Okay? So for resilience, what you want to do is make sure that if they're about to become a killer, because you're old enough to have had cells die due to amyloid and tangles, but there's really no infection there, right? That's resilience. So we want more housekeeping and less killing is the key. So what we did, and this is Duyan Kim's group in our lab, and this is the only complicated data slide I'll show, is we make mini human brain organoids. This is a paper punch sized well. Sometimes it's a little bigger well, like an inch wide. And there's gel in it that's like the brain. And we put in different types of stem cell derived brain cells, neurons, the microglia, the astrocytes, and they grow in this gel. The first version of this Alzheimer's in a Dish was funded by Cure Alzheimer's Fund 15 years ago when everybody thought it was crazy. And then we published in 2014 in Nature the first Alzheimer's in a Dish model that actually proved that amyloid does cause tangles. It was the first time there was proof that amyloid directly causes the tangles. And so we grow these, and what we do is we put in either regular centenarian microglia or just younger control microglia or microglia from Alzheimer patients, or we put in super ages centenarian microglia and we say, what do they do? And what we found, to our astonishment, was what you're looking at here is amyloid beta 42. A beta 42 is the one that really drives plaques, okay? And you're looking in the dish. The microglial cells were put in, either regular ones or from Alzheimer patients or the super ages centenarian ones. And you're looking at how much A beta is still there at 24 hours, 48 hours, 72 hours. And see these last two lines? These are super ages centenarians. And look how much A beta is still there. It's gone. And so this was really, actually, Du and I had a bet. Is the resilience in these centenarian super ages due to microglial cells that do a better job cleaning up the amyloid so you accumulate less over your lifetime? Or is it that they make the neurons more resilient, the nerve cells more resilient to the plaques and tangles and inflammation? And Du was betting on the neurons, I was betting on the microglia, and I won because it looks like the microglial cells really chomp up the amyloid much more than regular microglia from other people. So the question, that leads to a question, can we, the average person who's not a super ages centenarian, somehow figure out how to modify our microglia to prevent Alzheimer's pathology early in life and all your life, just like centenarian super agers? And so what can we try to do that? So what we did was we said, well, let's try to test what's already on the shelf in the pharmacy, FDA approved drugs, natural products that you can buy in supplements, although most supplements you don't even know if they contain it, but natural products are also available. So we took our Alzheimer's in a Dish model and the organoids, and we screened every known approved drug and 4,000 natural products. And the cool thing about these organoids is that, remember what I told you that on average, Alzheimer's pathology begins 22 years before symptoms, right? We get that whole process that can take 2 to 3 decades, from plaques to tangles to inflammation to nerve cell death, to happen in just 6 weeks in a lab dish. And that has made screening for drugs and natural products that can stop Alzheimer's pathology 100 times faster and 100 times cheaper than when we had to screen one drug at a time in mice, in Alzheimer's mice, where each experiment took a year and you had to do one at a time. Now you can do hundreds at a time. So over the past 10 years, with Cure Alzheimer's Fund funding for what's called 3DDS, 3D drug screening, meaning the three-dimensional model of Alzheimer's in a dish, the organoids, and we screen every known drug that's FDA approved and natural products and say what works. And our main goal was this: let's take those microglial cells that are causing inflammation and killing and get them to clear more amyloid instead. Turn the killers back into housekeepers, or try to enhance the housekeeping, try to diminish the killing. And this just shows an example of two natural products where when you put them together, okay, this is how much amyloid is getting cleared by the microglia. You can see that the first one does this much, the second one does that much. Together, they add the additive effect. And then we take these same compounds plus others, and we say, can we prevent the inflammation? Here we're measuring a cytokine called TNF alpha, which is part of cytokine storm and inflammation. And you're seeing its levels here. And then as you add these natural products, it goes down. So these are the types of things we're combining together to reprogram microglia to eat more amyloid and prevent the disease, blow out the match. But in case there's a wildfire, tame that wildfire, turn down the neuroinflammation. And we have 300 hits, 300 FDA-approved drugs and natural products that hit the pathology. It shows how they suss out here, which ones hit the amyloid and get the microglial cells to eat more amyloid, which ones stop tangles from forming, which ones stop the neuroinflammation. And we're making combinations of these to reprogram microglia, clear more amyloid, trigger less neuroinflammation. And our clinical trials of these combinations are ongoing at the McCann Center for Brain Health that I direct at Mass General, and with the company Active Pharmaceuticals, who's developing the gamma secretase modulator. So finally, in the last part of this talk, I want to get into what can you do now while we're waiting for these drugs to come online, these combinations to come online. What can we do now with lifestyle? And at the McCann Center, we developed the Brain Care Score. I think it will work if you point your camera at this code here. You can get to the website at the McCann Center that shows you the Brain Care Score you can take. It asks you questions about your numbers, about your lifestyle, about your social and emotional relationships. And then you give yourself a score, and the max score you can have is 21. And then what you do is when you saw that where you didn't have a good score, like I don't check my blood pressure enough, I'm not checking my BMI, I'm not eating healthy foods, I'm drinking too much alcohol, I'm smoking, right? As you start to correct that, you go back and take the test again, and this allows you to keep track of your brain care. So that's the idea of the Brain Care Score. But I came up with an acronym for how to take care of your brain some time ago called SHIELD. And I'm actually now writing a book. I just signed a book deal with Random House Penguin to write a book about SHIELD. Finally took the initiative to do that. The book should be out sometime around December of 2026 or beginning 2027, writing it now. But SHIELD is sleep. Every time you sleep, you're clearing your brain of plaque and other debris. This is when that clearance system, what's called the lymphatic system, turns on. And during sleep, you every time you go from deep sleep and REM or dream sleep, it's like a rinse cycle for the brain. You're clearing amyloid and other debris out of your brain. So the more sleep you can get, the better. Even a power nap where you have a little bit of deep sleep and some dreaming is a rinse cycle for the brain to clear out the amyloid and other debris. H is handling stress. Stress is a killer. Stress produces cortisol. Stress causes chronic inflammation. So, thinking about how to moderate stress, being more mindful, you might practice meditation. I include avoiding nasty people or spend more time with your cat. Just petting your cat can really alleviate stress. And normally my cat would be in here, but the door is shut, so I can't pet my cat right now. Interaction with others, loneliness, meaning being alone and stressed about it. increases risk twofold. So staying socially active is also very important. Keep in touch with friends and family. Call up an old college friend. Exercise. Okay, so exercise, you want to aim at about 150 minutes, maybe 20, 30 minutes per day of moderate exercise. What exercise does is it increases blood flow to the brain when your heart beats faster. And what we showed in our lab with Cure Alzheimer's funding is that with increased blood flow, the muscles release a hormone called irisin that goes into the brain and induces an enzyme called neprilysin that dissolves the beta amyloid. And by the way, those super aged centenarians have a ton of neprilysin. So one easy way to be a super aged centenarian, exercise. You'll make the same enzyme in your brain with exercise. And exercise also triggers the birth of new nerve cells that can reinforce the brain and growth factors to help those new nerve cells to grow and be healthy. So people ask me all the time, and I never know how to answer, how much exercise do I need? And I say, well, it's not really clear how much, aim at 150. But now a study just came out of Mass General, Harvard this past week that kind of answers that question. This is the Harvard Aging Brain Study that's been tracking folks for 3 years who are elderly, and you're looking at which ones go on to Alzheimer's or not. They're older folks. They're being assessed for brain health every year. And what was found in this study that was just published in Nature Medicine this past week is that cognitive decline was delayed by 3 years on average for people who walked just 3,000 to 5,000 steps per day. That's really not that much, okay? And 7 years, cognitive decline was delayed by 7 years in people who walked 5,000 to 7,500 steps per day. So people always say 10,000 steps, even just 5,000 to 7,500 per day, and you delay cognitive impairment by 7 years. So that's at least one answer for how much exercise. And sedentary individuals who didn't do any exercise had the fastest buildup of amyloid plaque and tangles in their brain and on the way to Alzheimer's disease. So that's exercise. L is learn new things. Basically, the more synapses you make every time you learn new things, the more you can lose before you get into trouble. As I said earlier, it's synapse loss that correlates with degree of dementia. Every time you learn new things, you're building up new synapses like money in the bank. It's another form of resilience you can have against the ravages of Alzheimer's pathology. And diet. I think diet is most important. I think, especially if you have the best diet is a Mediterranean diet, less red meat, more fish, plant-based protein, fruits, veggies, nuts, seeds, leafy greens. I've been a vegetarian since college. I try to stay mostly vegan, but sometimes a pizza has to have real cheese on it. Sorry. So I'm a vegetarian, not vegan, but sometimes vegan. Gut microbiome, yogurt, or probiotic. Granola is fiber-free microbiome, prebiotic. Antioxidants in fruits like blueberries or in green tea, dark chocolate, red wine, two very good things. And cut down on the fats, the sugar, the carbohydrates. Your brain stem, the oldest part of your brain, is constantly making you want fat, sugar, and salt, like what you get in a Big Mac, because that was for survival, right? When we didn't care how long we lived, and it still drives us instinctively today to want that extra piece of pizza. Okay, you got to just be mindful about controlling that. So I'll just tell you at the very end here now about a trial that we did with Dean Ornish. Dean Ornish is famous for his cardio diet that has helped so many people. And this was a small trial. It was just 20 weeks, 51 patients. So we had 25 who went on this intensive lifestyle intervention, 25 who didn't. And it was based largely on SHIELD. We encouraged 7 to 8 hours per night of sleep. We provided them with a device I created that's glasses that does flashing light with music that induces a meditative state, even if you don't know how to meditate, gets you down to what's called a theta frequency or meditation frequency. Interaction, there were group Zooms, 3 one-hour sessions per week, asked for 30 minutes of aerobic or strength training per day. Learning, there were weekly lectures on brain health so they could learn new things. But most importantly, a diet, whole foods, minimally processed plant-based diet was delivered to them on a weekly basis, 20 meals and snacks for the patient and their spouse. And these were the results. Cognitive decline was slowed significantly using the same exact cognitive measure that's so hard to hit that pharma companies have to use for drugs. This was used also for the new drugs that were just approved, Leqembi and Kisunla. CDR, some of the boxes, is the toughest one to hit. It's the bane of existence for pharma companies. It was hit. Okay, cognitive decline was slowed. And one of the components of this CDR, some of the boxes called CDR Global Score, it actually led to improved cognition, which we've never seen before with a significant p-value. The plasma A beta 42 ratio was changed with a significant p-value indicative of amyloid removal in just 20 weeks, mainly based on SHIELD. And gut microbiome health was improved, probably because of the diet, where the bacteria that are healthy for the brain versus those that are not were increased along with the metabolites they make. And then a 40-week extension where people stayed on this after the 20 weeks, these cognitive benefits were maintained and showed further improvement. So now Dean is doing a larger study with about 150 people to see if it can replicate this small study, but it's a great and very promising start that says here's the first evidence that maybe SHIELD, especially diet, can have an effect. So that's SHIELD, sleep, handling stress, interaction with others, exercise, learn new things, diet. This is SHIELD 2.0, just to end with. There's a little different, more like what you should and shouldn't do. Smoking, don't, S. H is hypertension and hearing. Check your blood pressure, check your hearing. If your hearing is down, you can't stay interactive with people. You start to isolate yourself. Infection, get your vaccines. Treat recurring herpes. If you get a cold sore once a month or more than 4 times a year, take an antiviral. Okay, herpes can trigger, as we showed, triggers amyloid in the brain. So manage your herpes. If you're getting cold sores, it's a sign that herpes is being activated, perhaps also in your brain. Moderate your alcohol intake. E is excess alcohol. L, lipids, check your cholesterol, especially LDL. Keep that in check. And D, dental hygiene. Floss regularly, get routine cleanings, because when we look in the brains of Alzheimer's patients post-mortem, we see more periodontal bacteria in their brains than in controlled brains. And we think the periodontal bacteria can help trigger amyloid deposition in the brain, because I didn't talk about it today, but my colleague Rob Moyer and I showed that amyloid beta protein is an antimicrobial peptide in the brain. It defends against bacteria. And bacteria, if they get into the brain, for example, from your gums, can trigger the formation of amyloid. The amyloid traps the bacteria to defend the host cell. So that's SHIELD 2.0. So I just want to say thank you to the Cure Alzheimer's Fund. If you want to support this type work or continue, these are all the different consortia, the Cure Alzheimer's Fund funds that are representative of all the different things they do, from tangles, gut microbiome, brain aging, how the microglial cells work, the role of ApoE, how do things get in and out of the brain. And this is just the tip of the iceberg of the many different incredible projects and incredible difference. No foundation has made a difference in this disease in terms of paradigm shifts or driving the ball forward to a cure more than Cure Alzheimer's Fund. No one's even close. And bang for your buck in terms of the dollars spent by Cure Alzheimer's Fund and the progress made outpaces everyone, including the NIH. And I'll stop there and thank you.
I hope you enjoyed this presentation of some of the cutting-edge research that Cure Alzheimer's Fund supports. I invite you to find a wealth of additional information on our website, curealz.org. Thank you for spending this time with us to hear about the science and progress that will bring an end to Alzheimer's. Please be in touch should you have any questions about the science we fund or how you can be part of the solution.